I'm making this video in response to a video that Talia made, talking about whether GMOs are bad or good. And I think, first of all, it's a fantastic video. I'm so happy that people are getting engaged with the debate, and I think it's really good. And it's something that I've been involved with since I was at university. So I was actually kind of anti-Monsanto, a little bit anti-GMO when I first started my biotech degree in genetics. And and then throughout my degree, I got more involved and more educated and felt more confident to start engaging in some of the topics that people were talking about. I think you covered heaps. There's a few little things I just wanted to talk about, which were just like small corrections in the language and terminology. So there was one thing you said at just over 2 minutes, which was talking about once you modify something, it's kind of permanent. So there are a number of ways we can modify stuff. And was one thing you said at just over 2 minutes, which was talking about once you modify something, it's kind of permanent. So there are a number of ways we can modify stuff. And when we're talking about modification, we're talking about modification as a technology generally. So when we selectively breed plants, under the current Australian legislation, it's not considered genetic modification, even though it is a modification of the genes. So one of the things that I wanted to talk about is a transient modification. So you can take something like a lettuce. I was lucky enough to work with a group in America called Miraculex, and they were doing transient modification of plants. So the way that they do this is they take a grown lettuce that hasn't been modified, they make its cells permeable to DNA, and then they dip it in a bath of DNA and pull it back out. This means this modification is carried on a plasmid, and a plasmid is a small circle of extra chromosomal DNA. So it doesn't actually change the genome in the plant the way that we traditionally think about it. So this doesn't get passed on to the progeny unless that happened to transfect a cell that would become a sex cell, and that plasmid was carried along. But selective pressures actually stop this from happening. So the same thing happens when we transfect groups of bacteria, for example. Across many generations, the plasmid doesn't get passed down and it eventually dies out. It's actually a problem for biotech. The other thing is that it doesn't necessarily always create a new species either. So the biological definition of species, we're generally talking about things that can't crossbreed and create fertile offspring. So sometimes you can create a new species, and we see this a lot in agriculture. So if we crossbreed two plants, a really good example of this is in orchids, but generally in crops, we can crossbreed plants that can create new species. But generally the small modifications don't create new species. And also when we do a thing called cisgenics, it doesn't create a new species either. So often, cisgenics is kind of the opposite to transgenics. So in transgenics, we're taking something like a gene from an animal, say. We might take a gene from a crab and try and put it into a tomato. So that would be transgenics because it's coming from a different species. But one of the things that we're really interested in is as you're talking about like drought tolerance, is we might find a drought tolerant species of wheat, and we might find a high yielding species of wheat, and we might try and transfer the drought tolerance genes to the same species of wheat to give that wheat the ability to resist drought and be high productive. So that's an example of cisgenics, and it's regulated slightly differently as well. You're also talking about insulin being a fantastic thing, and I agree. Biotech insulin is fantastic. We're actually running out of BioFoundry a project called Open Insulin, because even though the insulin has been made more readily available and lower cost, it's still not available to people in developing countries. So there's economic factors that sit behind the project itself. So we're trying to make that more open so anyone can make insulin. So that's one thing that people are quite upset with respect to genetic modification, is that it's in the hands of big companies, and through the biohacking movement, we try and stop that from happening by taking control of it as citizens. There's another thing I wanted to say is there's another really important crop which people are talking about. It's called mosaic virus resistant cassava. So we talk a lot about maize, and this is a staple across the Americas. Cassava is the staple in Africa, and it's been affected by a blight, and this causes it not to grow. And there's some not-for-profits that are going in there and trying to make a GMO cassava that's resistant to this, and they've had really good success doing that. And that's a combination of Bill and Melinda Gates Foundation, and I believe Monsanto is involved with that trial too. You were talking about it taking decades and decades and decades to get these crops through to the table. It's almost true, but it's more like years and years. So in Australia, if you did everything 100% right and you wanted to put this out on a farm, it's going to take you a minimum if you use every loophole available, minimum of 170 business days. So, and generally it's going to take you between 5 and 10 years. So that takes a long time. If you're interested in that, the Office of Gene Technology Regulator in Australia has all the documents available on their website. They're fantastic. They're really happy to talk to you about these things. Also, we're talking about this food being safe or dangerous and needing to have such long time periods to work that out. And that's really tricky. So we've served over 1 trillion meals with no one dying from GMO crops. And the hard thing is that opponents to genetically modified crops are going to generally say that we haven't proved it safe. The reason for this, you can never prove it safe. You can only prove that it's dangerous. So in this case, the burden of proof is on the other side to prove that the crops have a problem. Now, the same regulations don't apply to selectively bred crops. So there's actually been cases in the past 2 decades of people selectively breeding potatoes that are toxic, and they don't go through the same regulations and controls that GMO crops do. So in some senses, GMO crops are more likely to be safe than selectively bred crops. Now, on that note, I'm just going to add that we're talking about GMO labeling. So GMO labeling is a really tricky one. I don't actually agree with GMO labeling. The reason for this is that people who want GMO labels want to avoid it because they think that it's dangerous. Now, if you put the labels on there, they'll say, if it's so safe, why does it need a label? And then they're saying, if you aren't worried, then why don't you label it? So it's a catch-22 where if you do label it or don't label it, the opponents of GMOs are still going to be upset. And by putting GMO labels on there, it actually adds to the unnecessary fear around GMO crops. Okay, so on that note as well, we're going to go to ecological damage, which is another point that you brought up. So one of the responses from Monsanto to the potential problems that could be caused with GMO crops was to invent this thing called the Terminator Gene. Now, on the Terminator Gene, it basically created sterile crops so they can't reproduce in the environment. And one of the problems is people said, well, that means that we can't grow our own food because if we buy the seeds, we have to buy them from you again the next year. So you face this thing where you have a double where the things that would stop it proliferating in the environment, other opponents for other reasons are going to get upset around those measures too. Now, there's nothing actually wrong with the Terminator Gene. If you've watched Jurassic Park, you know that life finds a way. So you're not really worried about this because it's selected against in the environment. So on that note, it's probably not the greatest thing anyway. If you're looking for other ways of making it isolated to stop ecological damage, I would advise looking into things like making genes that are created with a left-handed double helix, so it creates a new branch of the tree of life. Physical separation, which you mentioned, and there's also another thing which is I wanted to add, which was you were talking about the types of crops that we had. One of them was the Roundup Ready crops. So one thing that I think we're not concerned enough about is the impact that glyphosate has on the environment, which is Roundup. So this affects the microbiota in the soil, and it also gets washed into the waterways. And I think that we should be a bit more mindful about how that affects the microbiota that exists in the water and also in the soil on farms. I don't think it's necessarily a huge issue, but it's something that we should be really thinking about when we're doing things like spraying this all over the continent. Also, you said that creating new species unnaturally is going to have some effect on the environment, which will change the ecosystem as seeds are spread. So all farming has an effect on the environment. Anytime we take a crop that wasn't grown in Australia, we place this in the environment and it has some sort of effect. So it's actually broader than including GMOs, and it's something we should be talking about, especially with respect to things like compartmentalization of the land and how that affects the Australian ecosystem. And finally, I'm just going to wrap up where you were talking about labeling. So we're saying things like soy. One thing that I got really interested in is in America, they have labeling on a lot of their products. But one of the things that is kind of contentious is that the product of a GMO is not a GMO. So if we, for example, grow a soybean and it's genetically modified, it's a genetically modified organism. The oil that comes from the GMO is not an O, so it's not a GMO. If I took soybean oil that had been made by a non-genetically modified soybean and I take a oil that's made from a genetically modified soybean and put them through a mass spec or a gas chromatograph, you can't actually tell the difference. That's because they're identical products. So the product that comes out of the refined product that comes out of the plant itself isn't a genetically modified organism, and this has huge implications for policy. Anyway, I want to say thank you again for making the video and getting involved with the debate, and I'd love to hear your thoughts on that as well. Thanks very much. Oh, hold on, I just saw that there's some comments. Yes, that's exactly right. Thank you very much for your comment, Caden. See you all again next time.